Welcome to Key Elements to Consider When Packaging Parenteral Products, presented by American Pharmaceutical Review and sponsored by Wheaton Industries. My name is Mike Auerbach, Editor-in-Chief of American Pharmaceutical Review, and I'll be the moderator today. Before we begin, I'd like to inform our viewers that this event will hold a live question and answer session at the end of the presentations. You can submit a question at any time using the Q&A box on your screen. Also, please note the resource box containing documents pertaining to this webinar that you can download. And now, allow me to introduce today's presenters. A member of the scientific community for 10 years, Jeffrey Reed earned his chemistry degree from the University of Delaware and his MBA from Goldie Beacom College. Jeffrey started his career working on the development of submicron particles for HPLC columns, specializing in several different chemical reactions. Jeff also worked as a product manager at Buki Corporation, specializing in lab technologies and techniques, such as pressurized solvent extraction, automated solvent extraction, and solid phase extraction. Currently, Jeff works at Wheaton Industries as a global market manager, where his main focus is finding solutions for customers in the pharmaceutical industry. Dr. Andrew Feilden is the Chemistry Operations Director at Smithers Rapra and Smithers Pira. He leads the chemistry groups at the Shawbury and Leatherhead facilities in the UK. The Leatherhead site specializes in food contact testing, while the Shawbury site carries out extractable and leachable testing, GPC analysis, and food contact testing. Andrew has been working in the field of extractables and leachables for over 15 years and has presented at numerous conferences. He is a scientific advisor to the IPAC-RS Container Closure Group, and he was the co-chair of the LC Material Group. Welcome, Jeff and Andrew.
Hello, everyone, and welcome. First and foremost, I want to thank everyone for joining today's webinar on key elements to consider when packaging parenteral products. It looks like we have quite a few live viewers on today's webinar, so Dr. Feilden and I will try to make it as interactive as possible. We'll do so by having a polling session halfway through the webinar, and then we'll have a lengthy questions and answer session at the end of the webinar. If we don't get to everyone's questions, our contact information will be on the last slide. Please feel free to reach out to us, and we'll get an answer to your question as soon as possible. I also want to note that the slide deck we're presenting on, along with 2 white papers, are attached to this webinar platform for you to download at any time. I'm going to start off with a quick introduction of the overall webinar, get into key processes such as particulate cleaning, depyrogenation, and sterilization. We'll have our polling session, and then Dr. Feilden will get into an introduction to extractables and leachables, provide you with some guidances, go over the effects of sterilization on extractables. We'll summarize everything up, and then we'll have our question session. This slide pretty much outlines the overall process that a primary packaging component undergoes when utilized for parenteral products. It starts off with a pre-sterilization process known as particulate cleaning. This consists of a series of USP purified water and water for injection rinses, and it's meant to reduce the amount of subvisible particles that can be found in a packaging component. The test methods are outlined in USP Chapter 788, titled Particulate Matter and Injections. Followed by this step is a depyrogenation step, which is meant to reduce the amount of bacterial endotoxins that can be found in a packaging component. This is usually done at high temperatures, usually around 250 degrees Celsius. The test methods are outlined in USP Chapter 85, titled the Bacterial Endotoxins Test. Lastly is the sterilization process. There are 3 main methods for sterilization: high temperature and pressure, chemical sterilization, and radiation sterilization. The test methods are outlined in USP Chapter 71, the Sterility Test, and we'll get into each one of these shortly into the webinar. But presuming that the packaging component meets or exceeds these 3 standards, it then can be utilized for a parenteral product. Now let's specifically get into particulate cleaning. Before we actually get into the process and variables, let's first define what is particulate matter. Particulate matter can be defined as any mobile, undissolved particle other than gas bubbles that are unintentionally found in the end product. They can be extremely dangerous when injected into the body and can cause several adverse reactions such as vein irritation, local tissue infarction, anaphylactic shock, and even death. There are two test limits around particulate matter. The first one is for visible particles, which are greater than 100 micrometers. The test limits are outlined in USP Chapter 1, which pretty much states that all parental products must be essentially free from visible particles. The second one is for subvisible particles, which range from 2 micrometers up to 100 micrometers. The test limits are outlined in USP Chapter 788, and this is specifically what we'll get into for this webinar. There are three types of particulate matter. There's extrinsic particulate matter, which is material that is not part of the formulation, package, or assembly process. Intrinsic, which is material from sources related to the formulation, package, or assembly process. And inherent, which is material expected from the drug substance and formulation components. Extrinsic particles are foreign from the process, whereas intrinsic particles are part of the process or product. And inherent particles are part of the formulation. Some examples of extrinsic particles could be fibers, skin flakes, hair, or ordinary dirt particles, whereas intrinsic particles could be silicone oil, rubber, or glass particulates. As stated earlier, particulate cleaning is a pre-sterilization process, and that's because you would never depyrogenate or sterilize a packaging component that had high amounts of particulate matter within it. It's performed in a HEPA filter washing and drying machine and usually consists of 4 major steps. The first step, the packaging component is introduced to sterile air blowing, which is followed by 3 USP purified water rinses, which is followed up by one last rinse by water for injections. Lastly, the packaging component goes through one last step of sterile air blowing. It's important to note that the water is heated to roughly 80 degrees Celsius to ensure there's no bacterial growth within it, and the water pressure is usually set around 5 bar to sufficiently remove the particulate matter that could contain inside of it. When comparing purified water and water for injections, it's important to note that they must meet specific ionic, chemical, and microbial requirements. Purified water can be processed by distillation, ion exchange, reverse osmosis, or other approved methods, whereas water for injections, the USP recognizes distillation or reverse osmosis, and the EP only recognizes distillation. For the bacteria growth, purified water must be less than 100 colonial forming units per milliliter, whereas water for injections, it must be less than 10 colonial forming units per 100 milliliters. The endotoxin count for purified water and water for injections must be less than 0.25 endotoxin units per milliliter, and the total organic carbon count for purified water cannot exceed 500 parts per billion. For water for injections, it has to be less than 0.5 milligrams per liter. There are 2 test methods for USP 788 particulate matter and injections. The first is a light obscuration particle count test, and the second is a microscopic particle count test. These are usually done in a 2-tiered approach, which means that if the light obscuration particle count test fails or the results are suspicious, then the microscopic particle count test is performed. Usually the microscopic particle count test is commonly used as an assurance test, which means the light obscuration particle count test is the premier test method for USP 788. And we'll get into reasons why with this slide. The light obscuration particle count test utilizes an electronic particle counter, A liquid sample is injected into the particle counter where it passes through a flow cell. This flow cell contains a light source and detector. When the particle passes through the light source, it's refracted, which then generates a pulse. This pulse then generates a calibration curve, which provides the operator with the amount of particles that pass through the light source and the actual size of the particle. Therefore, it's a qualitative and quantitative analysis. It's a strictly automated process, which is always paramount when you're doing quality assurance. Anytime you're utilizing automation, you eliminate subjectivity that could come from human error. So that's why it is the premier method for USP 788. The second method is a microscopic particle count test. This works as a sample liquid is filtered through a filter membrane with pore sizes less than or equal to 1 micrometer. The captured particles are counted at 100 times with a compound binocular microscope. It's a very operator-dependent method, as the operator is utilizing reference particle sizes to determine the particle sizes that were captured. So it's prone to subjectivity. However, it's very advantageous to utilize this method if your sample can cause emulsions, colloids, or if you're working with liposomal preparations. This slide pretty much shows the allowable or acceptable particle count test for USP 788. For the light obscuration method, particle sizes that are greater than or equal to 10 micrometers, small volume injections are allowed to have 6,000 per container, where large volume injections are only allowed to have 25 per milliliter. For particle sizes that are greater than or equal to 25 micrometers, small volume injections can have up to 600 per container, and large volume injections cannot exceed 3 per milliliter. For the microscopic method, particle sizes that are greater than or equal to 10 micrometer, small volume injections cannot have more than 3,000 per container, or 12 per milliliter for large volume injections. For particle sizes that are greater than or equal to 25 micrometer, small volume injections cannot have more than 300 per container, and for large volume injections, they cannot exceed 2 per milliliter. Now let's get into depyrogenation. As stated earlier, depyrogenation is a reduction of bacterial endotoxins. Bacterial endotoxins are made up of lipopolysaccharides, which are molecules found as part of the cell wall of gram-negative bacteria. If introduced to the body, they can cause severe fevers and even death from shock, so it's paramount to reduce these to acceptable amounts. This is done by exposing the product to high temperatures that are free from water vapor. The FDA requires a validated 3-log reduction in endotoxins from a starting concentration of at least 1,000 endotoxins per unit. Some typical cycles could be 250 degrees Celsius for 30 minutes or 200 degrees Celsius for 60 minutes. However, it's important to note that the process cycles are strictly dependent on the load variation. This can be done in a batch depyrogenation oven or a continuous tunnel. This slide compares the difference between a batch oven and a continuous tunnel. Note the pictures on the right-hand side of the screen. The top picture is of a batch oven, whereas the bottom picture is of a continuous tunnel. If you note the openings of each one, you can see that the batch oven is good with handling larger volume containers or containers that may vary in sizes, whereas the continuous tunnel may have bottle size constraints, but it is good for similar product pieces. It also combines discrete processes such as steam sterilization and depyrogenation, which means it supports in-house filling, and my next slide will really elaborate more on what I mean by this. Here are 2 flow paths. The top is of a continuous process. The packaging component is first introduced to particulate cleaning and then goes through a continuous depyrogenation tunnel. Since there's no human interaction after the continuous depyrogenation tunnel, it can then be pushed right out to be filled with the final product. With the batch process, the packaging component first goes through particulate cleaning, gets put into a batch depyrogenation oven. After this, there's human interaction with the packaging component, so it has to be sterilized. The packaging component comes out of the depyrogenation oven, gets put into a double autoclave bag, and then sterilized. After the sterilization step, there's no human interaction, as the human that's interacting with it is only interacting with the autoclave bag and not the packaging component itself. So it doesn't get contaminated. It gets put into a double heat sealed clean room bag, packed for storage, and then it's shipped out to be filled. There are 3 test limits for USP 85 Bacterial Endotoxins Test. The first is the gel clot method, the second is the turbidimetric method, and the third is the chromogenic method. Although these methods are different, they're based on the same fact that bacterial endotoxins will react with LAL. LAL stands for limulus amoebocyte lysate, which is an aqueous extract of blood cells from horseshoe crab. This slide shows a schematic of the LAL cascade. The endotoxin comes in and reacts with factor C. Factor C then becomes activated, which then activates subsequent enzymes within the cascade. Ultimately, it transforms the clotting enzyme to the clotting enzyme, which cuts the coagulagen into coagulin. The coagulin is a protein that actually will react with the endotoxin to form the gel clot, and that's the basis for the gel clot method. For the turbidimetric method, a turbidimetric assay is introduced to the reaction, which will cause turbidity with the actual reaction with the endotoxin and the coagulagen. And then the turbidity is measured, which is the basic behind this method. The chromogenic method introduces a chromogenic substrate, which is cleaved by the natural substrate, which then produces a yellow color. So you can see they're all based off the same reactions, but they are different in the last steps. The gel clot method works as follows. 0.1 ml of lysate is put in with 0.1 ml of a sample. It's placed into a reaction tube, heated in a water bath at 37 degrees Celsius, and then incubated for 1 hour. A positive reaction will show a gel clot at the bottom of the reaction tube. This is a very advantageous method because it is the reference method for USP and EP. It's very inexpensive and easy to perform. However, there are some disadvantages. For example, you're working with fixed incubation times. It's a very manual process which is prone to subjectivity. For example, if you flip the reaction tube too fast or if you move it too much, you can actually break the gel clot that formed at the bottom and not even notice that a gel clot formed. It's also prone to interferences such as vibration, pH changes, and temperature changes. It's important to note throughout this entire process that the pH must be between 6 and 8. When comparing the turbidimetric method with the chromogenic method, the turbidimetric method links the rate of turbidity to measure the endotoxins. Pretty much it's the turbidity versus the endotoxin of concentration. This method can be endpoint or kinetic, however, endpoint is not commonly practiced. The chromogenic method utilizes a chromogenic substrate known as PNA, which when reacts with LAL and endotoxins, it produces a yellow color. The darker the yellow color, the higher amount of endotoxins present in the sample. This also can be endpoint or kinetic. Lastly, let's get into sterilization. The meaning of a sterile product is that it's free from bacterial or other microorganisms. anytime you're talking with anything you're talking with sterilization, you always have to bring up the sterility assurance level, which pretty much means that the probability of having a contaminated product is 1 in a million, 10 to the minus 6. There are two test methods for USP 71. That's the membrane filtration method and the direct transfer. Dr. Feilden will touch a little bit more on sterilization in his section. But now let's get into the polling questions.
Thanks, Jeff. Let's take a look at our polling questions. We've got a few here to get through. So let's take a look at the first one. So our first question is, what is the preferred method for USP 788? Please respond by clicking on the box next to your answer and hitting submit on your screen. We'll give everyone a few seconds to input your answer. And I also want to remind everyone while we're doing that if you have any questions for our speakers, please enter them in the Ask a Question box on your screen, and we'll get to them during the Q&A session. So let's take a look now at the results. So almost 70% said light obscuration method, with 23% saying the gel clot method, and almost 8% saying the microscopic particle count test method. Jeff, do you have any comments on these answers, and let everyone know exactly what the correct answer is?
So the correct answer is a light obscuration method. That's what's outlined in USP Chapter 788, and that's mainly due to the fact that it is an automated process. The gel clot method is not for the USP Chapter 788. That's more associated with USP Chapter 85. So yes, the correct answer is B, the light obscuration method.
Excellent. Okay. Let's move on to our second polling question. And it is a true or false question. And the question is LAL the basis of all USP 85 test methods? So please select your answer and hit the submit button on your screen. We'll give everyone a few seconds to think about it and respond. And okay, let's move on to see what the poll results are. And the overwhelming majority said true. LAL is the basis of all USP 85 test methods. About 87.5% said that. Jeff and Andrew, any comments on this, the results of this polling question?
That's absolutely correct. And LAL is a linulous amoebocyte lysate, which again comes from horseshoe crab, and when reacts with bacterial endotoxin, it actually forms a reaction. So yes, that is correct.
Excellent. All right, we've got one more polling question for the audience. Let's take a look at that one right now. And our final question is, what pre-sterilization process must be done for injectable drugs? And our choices are particulate cleaning, depyrogenation, autoclave, or none of the above. And again, we'll give everyone a few seconds to choose their response. And as soon as everyone does that, we will take a look at the poll results. So I'll give you a few more seconds here. So now let's move over to the poll results. And the majority, 53%, said depyrogenation, followed by about 44% saying particulate cleaning, and a little more than 3% said autoclave. So Jeff and Andrew, if you could let us know what the correct response is and any comments regarding these results.
So that's actually a pretty tough question, but the correct answer would be A, particulate cleaning. Depyrogenation can almost be a sterilization step. So anytime you're talking about pre-sterilization, you want to introduce the packaging component to a particulate cleaning process because you would never want to, let's say, depyrogenate or sterilize a packaging component that had high amounts. So the particulate cleaning process will actually reduce the amount of visible and subvisible particles that could be part of it. Then it goes through the sterilization process, such as depyrogenation, and then depending if you're doing in-house filling or not, you would have to auto-sterilize it. So the correct answer would be A, but I could see where B could be a little controversial as well.
Okay. Thanks, Jeff. And now you can continue on with the second portion of your presentation. Thanks so much.
Thank you.
Okay, thank you. And now I'm going to cover more areas in terms of extractables and leachables, going into a few more details about exactly what they are, what are the guidances that are available, and just to summarize at the end on what the impact of sterilization on extractables with particular examples, so you can have a look at those. So the first key part is, what are extractables and what are leachables? There is normally quite a misunderstanding of what they are, and they're important because they're of where they actually come in the process and what you can actually do to them. So extractables, these are chemical species that can be released from the pharmaceutical packaging and or delivery system, packaging component, or any material that is in contact with the actual formulation under laboratory conditions. So it's using elevated temperatures, harsher solvents, different stoichiometry to try and pull out as much as possible from the material without damaging or degrading the material. You want to find out what is present, but not what you're actually forming because of that chemical extraction. The leachables, these are in all intents and purposes the chemicals or the species that the regulators are concerned with, because these are the chemical species that end up in the product under normal use. So these are all the chemical species that the patient will be exposed to. So just in terms of clarity, extractables are possible impact, because when you do the extractable testing, you're trying to pull out as much as possible, but you're making sure that you pull out as much as you can, but they may not end up in the product, because leachables, these are what actually do form. So the leachables are actual impact. So just as a reminder of what object you test, extractables, you test the material or the component. The leachables, you test the final product, and that means testing from all the way through from initial manufacture all the way through to end of shelf life of the product, because typically leachables take time to form. Therefore, the maximum level of leachables are typically at the end of shelf life of the product. And just as a nice example for the difference between extractables and leachables, you test the materials as extractables and you test the product for a leachable. So this is a diagram that works, that shows what could happen in a container closure system. So you have a label with adhesive and inks. You have the primary pack interaction. You can actually have leachables that have come from in-process manufacturing systems. And finally, you can have secondary packaging or environmental issues. There were a number of product recalls in 3-4 years ago that were related to the biocides that were used to treat wooden pallets. And these biocides from the wooden pallets actually ended up in a number of products. So Johnson & Johnson had a big Tylenol recall simply because of that. So you can see there are multiple sources of leachables that can actually come into the product. There are multiple sources, and it becomes more complicated when there are more materials in devices. So from just a simple carton like that all the way through to syringes that may have a plastic barrel. You've got glue with the needle in place, and you've also got maybe an elastomeric plunger, glue or ink on the outside, labels, all kinds of extra things. So what are the regulations that are out there? There is nothing actually specific to do the testing for extractables and leachables for every single device. There are guidances, and these are things that you should follow. You don't have to follow. However, with them being authored by the regulators, it's something that you should follow because they've authored those, so they're the things that you need to follow. So there are There's a wide range of landscapes in terms of the regulatory. So we have the FDA, European Medicine Agency. There are also a number of working groups. So the most common, the PQI, the Pharmaceutical Quality Research Institute, USP, ASTM, ICH. Then there are also some industry groups, so ELSI, the Extractable and Leachable Safety Information Exchange, BPSA, Bioprocessing Systems Alliance, BPROG, the Bioforum Operations Group, and others exist. But this just gives you an idea of what is out there, what is actually available, and a number of those groups do interact. So people that are involved with the PQI, there are authors from the FDA, Health Canada are involved in that group, just to name a few. So just looking back at what guidance was out there, a lot of the guidances around extractables and leachables started off from meter dose inhalers, so anything that was administered to the lung, because these were devices that had a large amount of elastomeric materials that were involved and that were in contact with a nice organic solvent for a prolonged period of time. So the chance of something leaching out into the product was high, and actually being delivered to a damaged organ was also high. Therefore, the regulators became involved and wanted to improve the safety and quality of the componentry. So you can see things came out in 1993. We have in 1998 the FDA MDI DPI draft guidance. It is still draft guidance, so when that will ever be seen as finalized, I don't know. But we're 18 years on and it is still draft. So we have other guidances that came out. So we've got ISO 10993, aspects of USP, all those exist. So there was a range of guidances available. We then move on a number of years, 2007. Probably the key document there was the PQI Safety Thresholds and Best Practices for Extractables and Leachables in ONDPs. So these are orally inhaled and nasal drug products. So this was the first time that the regulators and others came up with a line in the sand of how low do you need to go to detect your extractables and leachables, because the number of chemical species that can come out from plastics or rubbers is very, very high. So at what point do you stop looking? You can spend more and more time looking for these chemical species that come out, but are they potentially harmful to the patient? So the PQI came out with that guidance that gave the line in the sand, at what point do you stop looking for it? And then coming points about the PQI, because of its importance. What it didn't do, it doesn't suggest specific test methods and acceptance criteria. So it doesn't say you must do this and then everything will be okay. It gives best practices as long as it is scientifically justifiable. Some of the key points it goes is using multiple solvents for your extraction, multiple analytical techniques. It does give the limit of how low to go down to. It also wants you to look for batch-to-batch uniformity to make sure that this batch is as good as the next batch, and it's all about patient safety. All the guidances around extractables and leachables are all about patient safety. Doing the testing will never make a product any better than any one other. It will just make it safer. Another key point that they came up with, correlation of extractables and leachables. Because as I said earlier on, leachables takes time to form. You can do your extractable testing very, very quickly. You can take your sample, stew it up in a solvent, then analyze it and find out what chemical species would come out. So a lot of the testing around extractables and leachables is seen as extractables is understanding the risk or carrying out risk mitigation, because the extractables are what could come out. So by carrying out the extractable testing early, it gives you a chance to either deselect that material or know that the material that you've selected in your device is likely to be okay. So it's making sure that when you test the product that it is as safe as possible. And just as a comparison, what's coming up, this is the difference in size scope of the EMA guidances. I should have said that the PQI document runs to 272 pages. And just on the screen, this is the summary of the EMA, so the European guidances on extractables and leachables. It doesn't go into a great deal of detail, so it's just a small amount of information. So the difference between the US and the EU is quite stark. And in the last 2 to 3 years, there's been a number of guidances that have come out. We have the latest USP 1663 and 1664 assessment of extractables associated with pharmaceutical packaging delivery system, and the assessment of drug product leachables associated with pharmaceutical packaging delivery systems. Both these guidances are have been taken from the PQI recommendations, and for those of you that are familiar with the USP system, numbers above 1,000 are recommended and those below 1,000 are mandatory. So these are recommendations, but it would be a recommendation to certainly follow. And as I've just said, what is E&L testing for? It's all about understanding the risk and reducing the risk to an acceptable level, because doing the testing will not make the product any better. It will not differentiate the product from a competitor's. It will just make the product safer for the patient. So a lot of the activities involved are around a risk-based approach for safety assessments. But how do you actually determine if the material is safe for its intended use? And it depends on a range of factors. Route of administration. Primarily, the highest degree of concern is associated with inhalation products, typically because they are in contact with a nice organic solvent for a prolonged period of time, and they're typically being administered to a damaged organ. We have things around toxicity classification, the effect of the contact materials. Are you going for small or large volumes? There can be a difference depending on the products administered. You can go from an asthma inhaler that may deliver 25 microliters at a time to a patient, or you can go all the way up to dialysis bags that may be delivering 75 liters to the patient. That's a huge difference in size and magnitude of the amount of material per dose that the patient would be exposed to. What is the dosage form? Is it an aqueous? We all know that water is a very poor solvent, so if you're administering something with water, the likelihood of something being in the water is quite low. But there are a number of drug products that require surfactants to help solubilize them. By solubilizing the active pharmaceutical ingredient, you're also enhancing the chance of solubilizing extractables or leachables. One of the questions is, what is the patient population? Is it to children, or is it an oncology product that you're giving to cancer patients? How often are you going to deliver it? Is it just going to be once, maybe once a year? Or is it something, maybe an injection or an inhalation product will be taken daily for the rest of their life? So the duration can play a big part. And there are two main aspects in terms of what is happening with extractables and leachables. Are they directly toxic, i.e. could cause harm to the patient, or are they going to cause drug efficacy issues? So will it have an impact on the potency of the drug? And this is of particular concern with biological molecules, that the extractables can actually interact with the large molecules, the proteins, and cause a degree of problems, maybe aggregation, folding, generally reducing the efficacy of the actual active ingredient. And this just summarizes, taken from the USP 1664, which is produced by the FDA, of which products have the highest degree of concern associated with the route of administration and the highest likelihood of packaging component dosage form interaction. So as I mentioned about inhalation, but also the injection solutions for injection are also very high up there. So they're in the next box that are concerned. And it's the question that the FDA has also produced in conjunction with the PQRI, a similar type of document for the inhalation, but now for PODP, so for parenteral and ophthalmic drug products. So that shows the state of the nation, as it were, of where there is degree of concern. At the minute, anything producing oral tablets, capsules, there is little risk and little regulatory concern about those. So what I'm now going to do in the last 2 or 3 slides is just highlight the importance of thinking about sterilization and the selection of materials. So we've already heard from Jeff about typical routes of sterilization. These can all have a potential impact on the level and type of extractable species that are seen. So some of the choices can depend on which mode of sterilization to use, but also which materials. And these are just some random examples. So looking for headspace GCMS analysis, so we're looking for the very volatile extractable species, using polypropylene as the material. So we irradiated the polypropylene and we also autoclaved it. And you can see from the plot the amount of chemical species and the number of them that we're seeing from the irradiated sample is very, very high. Polypropylene is not something that we typically want to irradiate. Because of the actual process, you do tend to see more volatile species, hence seeing a different analytical approach to see those with the headspace analysis. Another example. So this time we've taken a polycarbonate. We're doing the GCMS analysis, so rather than the very volatile species, there are semi-volatile. And the difference in the chemical species between the irradiated or autoclaved are negligible. There's a small amount more on the irradiated sample, but those are really down in trace levels. And again, so with the sterilization and the analysis, we've actually had to do a different analytical technique. So these are the areas that I haven't had a chance to go through in the webinar and go, okay, what are the approaches to the analysis of extractables and leachables? So, and the final example, we're doing LCMS analysis, so we're looking for the semi-volatile to non-volatile species. And this time we're seeing a different distribution in terms of what chemical species are seen between the irradiated and the autoclaved. And all the ergophos 168 chemicals are antioxidants. So the antioxidant is actually breaking down, and depending on the route of sterilization, in different ways. And you may think, well, why should I add an antioxidant? And as the final slide, antioxidants are there to prevent the degradation of polymers, typically by sterilization if it's light, heat, irradiation. So this is just an example of showing what actually happens, and the antioxidants that you saw degrade were actually doing their job to prevent the polymer from falling apart. So if you didn't have the antioxidants present, the whole polymer would have fallen apart and you wouldn't have had the material in question to act as the container closure system. So as the final slide, it's what you've heard today is we've given an overall scheme for injectable drug products. So you've heard about particulate cleaning, depyrogenation, sterilization, and aspects around the final packaging filling of those. So you've also listened about parenteral products, must meet or exceed the various USPs 788, 85, and 71. You've now heard me go over a very brief introduction to extractables and leachables, and just highlight the importance of thinking about the type of materials used if they require sterilization, and the actual choices in modes of sterilization you should pick before carrying out that testing. So thank you for listening, and we'll now progress onto the Q&A session. And just as a reminder, if you can use the Q&A box at the bottom of the screen to type in your questions.
Thanks, Andrew and Jeff. As Andrew just said, it's now time for our Q&A session, and you can submit any questions for either of our presenters in the Q&A window on your screen, and we will try and get to as many questions as possible. So let's take a look at our first question. And the question is, please discuss the disadvantages of both the continuous process and batch process of the depyrogenation process. Which process do you recommend?
So I can answer that, Michael. I would say the biggest advantages of the batch oven is that it's advantageous to use when you're working with larger components, or pretty much components that have varying sizes. So maybe you're depyrogenating a 1 liter bottle versus in a 500 milliliter bottle at the same time. So it's very good on applications such as those, whereas a continuous tunnel, that's good to utilize because you have less interaction with less human interaction, and anytime you have that, you have less chances of contamination. Now, which one would I prefer? I mean, it's really dependent if you can support in-house filling. If you can support in-house filling, then the continuous tunnel is definitely the way to go. Now, if you can't support it, or if you don't have it, then you would have to go with the batch oven.
Okay, great. Thanks, Jeff. Let's move on to our next question. And the question is Wheaton known for the services you presented on, such as depyrogenation, sterilization, and particulate cleaning?
So I can answer that one too. So Wheaton is known for providing primary packaging components to the pharmaceutical industry. What really differentiates Wheaton is the fact that we can offer ready-to-use products. And we do that through our premium services line, which consists of those following processes, whether it's particulate cleaning, depyrogenation, and sterilization. So, yes, we've been around for 126 years, and we're capable of doing so.
Thanks, Jeff. We've got another question here. This refers to slide 38, and the question is, was sterilization done in the samples from slide 38 using E-beam or gamma irradiation?
They were all done with gamma irradiation at 77 kilograms.
Okay. All right, let's move on to our next question. And the question is, when do you need to use USP pyrogen testing?
So anytime you're filling a parenteral product or a product that's meant to be injected, it has to meet acceptable limits for the reduction of bacterial endotoxins. So that process has to be validated. Any process you're working on, whatever oven you're working with, has to be validated, which means then you would have to prove it through those USP test methods.
Okay, thanks, Jeff. Let's move on to our next question. And the question is, what is the best way to characterize particulates found by light obscuration or microscopy?
Okay, so USP Chapter 1 really defines visible particles. For any particle range that is within 1 micrometer up to 100 micrometer, you have to utilize these two methods. What I can really do is elaborate more on the visible versus subvisible particle sizes. In summary, it's defined of a visible particle having any size greater than 100 micrometers, whereas subvisible particles have less of a size from 2 to 100 micrometers. However, it's important to note there is a visible gray zone in between 150 micrometers to 225 micrometers. So it's due to subjectivity from the inspector. So anytime you're really working with particulate matter that is subvisible particles, you would have to utilize these two methods.
Okay, thanks, Jeff. Let's move on to another question, and it is, can you briefly explain microflow imaging?
Dr. Feilden, do you have a little bit on that?
No, I don't know anything about microflow imaging. Sorry.
What we can do is, if you take that offline, maybe we could shoot us an email, and then we'll definitely look into it and try to find a little bit more of an answer for you.
Okay, that's a great suggestion. Let's move on to our next question, and it is, what is the difference between sterility and container closure integrity testing?
So the sterilization step has to be done with any primary packaging component, so it is the closure as well. So it has to be done, and a primary packaging component is pretty much defined with anything that is in direct contact with the end product. So the sterilization step would have to occur for both. Hopefully that answers what's being asked here.
Okay. I have another question here. I think this might be for you, Andrew. Do leachables impact chemical drug stability rather than biologicals?
The answer is, unfortunately, it depends. So it depends on the nature of the leachables. Are they particularly reactive? One of those common ones may be something like formaldehyde, and its interaction with certain drugs, it can cause them to degrade. So you increase the chance of finding sort of degradation products. So it can affect their stability in a similar way that biologics, not all leachables will affect biologicals in the same way. Certain leachables will do it for certain biologicals. So unfortunately the answer is it depends, and it's nearly on a case-by-case basis on what are the leachables and what are the actual active pharmaceutical ingredients involved.
Thanks, Andrew. I think I've got another question here for you. And it is, can you comment on the potential impact of SIP and CIP processes on extractables and leachables?
I'm not 100% sure what SIP or CIP processes are, so I would be guessing on what the three-letter acronym stood for. I'm not familiar with those. So
yeah, I believe they're referring to either like steam in place and clean in place processes.
Okay.
I guess for yeah.
So it has the potential. If you're putting any energy into a process, the likelihood of something coming out increases. So if it's anything involved with plastic or elastomeric, polymeric materials, the likelihood of something coming out increases. there was one FDA reviewer that went through a thought process on polynuclear aromatic hydrocarbons, so known carcinogens, that were coming out from or could be present in any system. And his thought process was something is manufactured involving moving parts. Those moving parts need to be lubricated. You typically use a hydrocarbon oil to lubricate those. The oil gets hot, and by the oil getting hot, polynuclear aromatic hydrocarbons could be formed, so you need to test for those in all systems. The likelihood of something like that happening is very, very small. But if a regulator asks you a question, it's easy to answer. You go ahead and answer it.
Thanks, Andrew. I've got a question here for Jeff. Regarding LAL testing, if a sample has a lot of protein, I have found that they may cause false positives or even false negatives depending on how they are cleaving or interfering with the assay. How can I overcome this?
One way to overcome it is through heat activation. What you can do is heat the sample to roughly 70 degrees Celsius for 5 to 15 minutes in a water bath, then do further dilutions on the inactive sample, and then test from that point moving forward.
Okay, thanks, Jeff. I've got another question here, I believe, for you. You provided the preferred methods for particulate cleaning and depyrogenation. Can you provide the preferred method for sterility testing?
So the preferred method for sterility testing is the membrane filter. The membrane filter sterility test.
Okay, great. Thank you. And let's move on to our next question. Question is the use of biological indicators required with each batch of drug that is terminally sterilized by autoclave or depyrogenation? Jeff, maybe you can take a shot at that?
Yes, the FDA requires BIs for both methods.
Okay. Let's move on to another question. Question is, siliconization of rubber stoppers will have an impact on will siliconization of rubber stoppers have an impact on leachables and extractables? Is there any acceptable value that should be limited to?
First part, the question, yes, siliconization will have an impact on leachables and extractables, and the acceptance limit is depending on the dose that's actually delivered to the patient. So the level of leachables depends on the dose. So if you're giving, say, a device 1 ml a day to the patient, the limit around the leachables are used of something that's called the detection level that's required is around the analytical evaluation threshold, which is set at 0.15 micrograms per day. So that's the level at which a leachable and extractable needs to be identified. So once you've identified those chemical species and they're above the 0.15 micrograms per day, you then need to talk to a toxicologist, and they can set an acceptance level of, is it going to be harmful to the patient? So this could be in the microgram level or the gram level or beyond. So a lot of the testing for the extractables and leachables is around the identification, so a toxicologist can assess, are these levels safe?
Okay, great. Thanks, Andrew. I think we have time for one more question. This one, I think, is for Jeff. Is the use of biological indicators required with each batch of drug that is terminally sterilized by autoclave or depyrogenation?
Yes, it is required for both, for each batch of drug.
Okay, excellent. Thank you very much. So I'd like to thank both of our presenters for their answers during the Q&A session, and unfortunately that's all the time we have right now. Any questions that we were not able to address here live will be answered by our presenters via email. We'd like to thank both Jeff and Dr. Andrew Feilden for sharing their knowledge with us today, and also offer a special thank you to Wheaton Industries for sponsoring today's event. Please keep a look out for an email containing a link to view this webinar on demand. Thank you everyone for attending, and have a fantastic day.